Good morning, everybody, and welcome to the fourth Grand Rounds lecture of this year. So this is a collaboration with the Shanks Pathology Foundation and the Pathological Society of Great Britain and Ireland, and with the generous sponsorship of the Shanks Foundation, we're able to organize these lectures and have an overview of various different cancer and biological studies. Today it gives me great pleasure to introduce Dr. Bárbara Hernando, who is a junior postdoctoral leader in computational oncology, and she's based at the Spanish National Cancer Research Center. Her research focuses on uncovering the mutational processes that shape tumor evolution and therapy resistance. She has a very impressive, albeit short, career in that she did her PhD at University in Spain before she did her first postdoctoral period in the Cancer Genome Evolution Research Group at UCL. She joined Dr. Jeff McIntyre's lab at the CNIO, and she's currently a visitor research scientist in Dr. Sarah Aitken labs at Yale School of Medicine and in the Department of Pathology. So without further ado, I'd like to hand over to Bárbara, who will speak for approximately 45 minutes. And please, everybody, if they could note the Q&A, we will try to address as many questions as we can towards the end of the hour. So thank you very much, Bárbara. I give the lectern and microphone to you. Thank you.
Thanks, Sarah, for the kind introduction. I'm going to share my slides. Okay. Okay. I think that now it's working, isn't it?
Yeah, that's great. Thank you.
Okay, perfect. So hi everyone. Thanks. First of all, I would like to thank the organizer for inviting me to give this talk today. It's really a pleasure to be here. And well, as Sarah said, I'm working at the Spanish National Cancer Research Institute in Madrid. And in my research group, we are mainly focused on trying to understand chromosomal instability in order to tackle these aggressive and complex tumors in a more effective and rational manner. So 80% of all cancer tumors have chromosomal instability. And this is a hallmark linked to metastasis, poor prognosis, and therapeutic resistance. What I refer about chromosomal instability is this type of tumor genomes. In this plot, you can see in the x-axis the chromosomes order, and then in the y-axis the number of copies of the different regions in the genome of this tumor. Normally, a normal sample has 2 copies of each chromosome, so a flat line at copy number equal to 2 will be represented here. So now you can really realize about the huge complexity in the tumor genomes that these tumors with chromosomal instability have. So compared to stable genomes, tumors with complex genomes with this chromosomal instability have a lot of copy number alterations. And the point is that the current approach of precision medicine is mainly focus on the detection of an alteration in a single gene or protein. And this works really well for stable genomes. The problem is that this huge diversity of the mutational landscape found in tumors with chromosomal instability limits our ability to personalize treatment. This is because normally we found driver alterations in different driver genes, and each one of these alterations is linked to a different targeted therapy. Therefore, treatment selection is a really difficult task, and that's the main reason why tumors with chromosomal instability are normally treated with cytotoxic agents. So the point here is that all these patients are treated in the same way. And this is because we don't have a good biomarker to select the optimal treatment for each one of those patients. So with this challenge in mind, in my research group, we are trying to understand the mechanisms driving a normal sample, as the one in the top, to evolve into a tumor with high levels of chromosomal instability. Because if we have this information, we can actually avoid tumor progression, treatment-resistant metastasis, and ultimately death. So in my talk today, I will present you 3 different stories. In the first part of my talk, I will show you how we can infer the mechanisms of the mutational processes driving chromosomal instability, and how we can use this information to predict treatment response. In the second part of my talk, I will present you a new approach that we use to forecast or to anticipate the acquisition of new driver alterations and therefore tumor evolution using these type of tumors as an input. And then in the last part of my talk, I will be presenting you a new approach where we use single-cell DNA sequencing to understand or to dissect these mechanisms that are actually active in a tumor and are driving the accumulation of new copy number alterations. So let's start with the first part. So in this first part, I will show you a new approach that we developed and published a few years ago, where we can dissect the mechanisms that are actually driving chromosomal instability. And our approach is based on the concept of mutational signatures. So basically, what we are doing is to try to look for different genomic footprints or patterns of copy number alterations in a tumor genome. This is mainly similar that one it's done in a crime scene. So in these tumors, what we see is different patterns. So it's a mix of different patterns of footprints. Then we have developed an approach that is able to unmix these different footprints into signatures. And then each one of the signatures can be linked to deficiencies in specific pathways that actually this information can ultimately use to select the optimal treatment for each one of those tumors. So in our work, what we did was to dissect all the footprints or all the copy number alteration patterns seen in the TCGA. And after mixing all these patterns, we developed a compendium of 17 signatures of chromosomal instability. Then the next step was to integrate all different orthogonal and additional data from the TCGA and from other cohorts in order to understand or to link the etiologies of the mutational processes driving these different signatures of chromosomal instability. So after doing all this integration of data, we have our compendium of signatures where we that these signatures are representing different types of chromosomal instability. For example, we have signatures linked to chromosome segregation errors or mitotic errors. We have signatures of impaired homologous recombination, signatures of linked to whole genome duplication, to replication stress, impaired non-homologous enjoyment. So the idea is that we have all these patterns that are linked to putative etiologies, so to deficiencies of pathways that are actually causing or shaping the tumor genomes. But of course, all these etiologies that we propose were by integrating or linking different layers of orthogonal data. So now we are working on validating all these signatures in vitro to have this causal link. For that, we started by generating an engineering model where we knock out BRCA1 and BRCA2 in order to induce impaired homologous recombination. Then we derive copy number profiles from these induced models and explore the patterns of copy number alterations that those cell lines actually acquire. And interestingly, what we see is that all these copy number alterations, this pattern of new copy number alterations, have some features that are actually very similar to the signatures that are associated with impaired homologous recombination in our compendium of signatures. So we actually can have a causal link between the deficiency of impaired homologous recombination with the signatures that had been associated with impaired homologous recombination in our previous work. Then we also induce other models of chromosomal instability using reversing and doxorubicin. Actually, reversing is a drug that is associated with the production of replication stress and also micronuclei. And what we see is that the copy number alterations associated with this reversing are linked to the signature that have replication stress associated signature. And the same with the reversing. The reversing is a drug that causes mitotic errors, and we see a similar pattern of copy number alterations linked to chromosome desegregation associated signature. Now, of course, these are 4 different signatures or 4 different etiologies, but now we are working on expanding these in vitro models of induced chromosomal instability, but knocking out different genes that are linked to DNA damage responses. And the main goal of this project is to validate the causal mechanisms for our compendium of 17 chromosomal instability signatures. So once we have this technology that is able to dissect footprints or patterns of copy number alterations and dissect that into signatures, the next step was to test if our signatures actually have utility in the clinical setting. So our hypothesis is that we have all these signatures that are representing different types of chromosomal instability, and these signatures can actually be used as genomic biomarkers to predict treatment response, and therefore to be able to select the optimal treatment for these patients. I'm going to focus today in a project that we published last year, where we use our signatures to predict resistance to 3 different cytotoxic chemotherapies: platinum-based chemotherapies, taxanes, and anthracyclines. So in this work, what we did was to emulate randomized clinical control trials by applying our signatures to classify the patients as sensitive or resistant to one of these 3 chemotherapies. Then what we did was to compare the time to treatment failure between those samples that were in the experimental arm, therefore treated with the specific chemotherapy that we were interested on, and compare those to the control arm, to those patients that were treated with the standard of care. The good point of working with chemotherapies is that these drugs have been used during the last 20 years. So we can use real-world data, retrospective real-world data, to emulate all these randomized clinical trials without the need of starting a clinical trial, a prospective clinical trial from scratch. So for that, we use data from the PCGA and the Harvard Medical Foundation data sets. We perform a curation of the genomics, clinical, and therapy response data, and ended up with a cohort of 840 patients that have primaries and metastasis and metastatic samples. And we performed these randomized controlled trials across 4 different tumor types. So just let me show you an example. In this case, I'm showing you the emulation of a clinical trial to predict resistance to taxane in relapsed ovarian cancer. What we did first was to apply this inclusion and exclusion criteria that is done in all the clinical trials. And after that, we apply our biomarker to classify the patients are resistant to taxane or sensitive to taxane. And then we compare the time to treatment failure between those patients treated with taxane as with a standard of care. And what we show in our results is that actually our biomarker is predicting those patients that are resistant to taxane because these have a shorter survival time compared to those that were predicted resistant but were treated with a standard of care. We performed that simulation across 4 different tumor types and primary metastatic. This is a table summarizing all the results that you can find in our publication. And the main conclusion for that is that our signatures can actually inform about the use or the benefit of using chemotherapy at different tumor types and patients across primary, metastatic, and 3 different chemotherapies. So just to summarize this first part of my talk, I show you that we have developed an approach to extract STING signatures, and these signatures are linked to specific mutational processes and therefore can be used to identify tumor vulnerabilities. And also that these signatures are useful biomarkers for tailored administration of chemotherapies. And the good thing of this test that I didn't have time to go through in detail is that we can detect the prediction of these 3 chemotherapies in a single genomic test. So you only need one test to have all the signatures and therefore all the biomarkers. It is also tumor agnostic because we apply that to 4 different tumor types and is compatible with multiple sequencing platforms and sample types. So in this first part of my talk, I already showed you that we have these signatures, that these signatures are useful to understand the biology of a tumor with chromosomal instability, and that we can use this information to predict treatment response. But then the next step, what we thought is that, can we actually use this biological information to forecast or to predict what is going on, what is going to happen after a time in this tumor? So can we actually forecast tumor evolution? And this idea was inspired in previous work where these words show that tumor evolution is actually deterministic. So it is there is like an order of events that are normally acquired in this tumor evolution. And this divergence between tumors and others is because this order can be changed, but actually is quite deterministic rather than stochastic. So we developed this approach based on classical evolutionary theory. For a mutation to be fixed in a population, what we need, or selected in a population, we need that the mutation occur, and also that this mutation is selected. So then the mutation will be expanded in the tumor population. So for that, we need a mutation process that is generating this mutation. We need this fitness advantage of the alteration. So the alteration should be positively selected. And then the clone that is harboring this alteration will be expanded in the tumor population and will be actually detected at pool level in a diagnostic set. So because this modeling of tumor evolution is quite like a huge task, we started by forecasting oncogene amplifications. And our model, our forecast model, it's combining the driver-specific mutation rate. So what is the mutation rate, the proportion, like the chance of this driver is amplified? Also, the selection coefficient of this driver amplification. If this oncogene amplification occurs, it will be selected. And then we compute a probability of this driver to be actually amplified. For the driver-specific mutation rate, what we do is to use our SIN signatures. So from a tumor sample, we input this tumor sample and we dissect the different mutational processes operating in this tumor. And then we combine that with a pre-computed mutational processes operating in the specific driver or oncogene that we are interested on. And this was computed. I'm not going to go to specific details, but actually these driver-specific mutational processes was computed using 7,000 tumors across 33 tumor types and a huge number of driver genes of oncogenes in this case. Then we combine this driver-specific mutation rate with the selection coefficient. So we use GISTEC, that is a computational approach, to know if the driver alteration is selected or not in a specific tumor type or cohort. In this case, we apply this driver selection coefficient across the different tumor types, the 33 tumor types. We train that using 10,000 tumors across these huge amounts of driver genes that we selected for, so these oncogenes. And once we combine these two information, we have this probability of amplifying a driver alteration. And we apply or we train an optimal threshold to classify the patients as having high risk of a driver alteration and a low risk of a driver alteration. So in a clinical setting, what we have is an input that is a tumor sequence where we derive copy number signatures and copy number profiles. And the output will be if this patient have high risk of amplifying the specific oncogene or low risk of amplifying this oncogene. So once we have this forecasting approach developed, what we did was to actually assess if it works. So for that, we use longitudinally collected tumor pairs. We apply our forecasting framework in the early sample. So in the sample at diagnosis, where the amplification is not seen or it's not there. And then we classify the patients as having high risk or low risk of acquiring the specific amplification. And we use the second time point to detect if actually the oncogene is amplified or not in this second time. And for that, we use 3 different use cases. And the results in all of them so very promising. So we are able to actually classify correctly the samples that will acquire this specific amplification in AR amplification in prostate cancer, H1, H3B amplification in lung cancer, that it's an alteration that is normally only seen in metastatic lung cancer, and then MET amplification in non-small cell lung. And you see that the AUC values in all of these cases is quite high. So here we are confirming that our forecasting framework works, but it's clinically useful because that is our main goal as a research group. So for that, we apply our forecasting approach in 2 different use cases. Because of time, I'm just going to focus on showing you the results of the first use case, where we forecast osimertinib resistant in non-small cell lung cancer via MET amplification. So as you might know, EGFR mutant non-small cell lung cancer patients are normally treated with osimertinib at first line. or EGFR inhibitors of guidelines. It works really well. The problem is that most of these patients develop resistance in a time frame. And here in this picture, you can see all the different alterations that can drive to resistance to osimertinib. One of these alterations is MET amplification. And actually 15 to 30% of the patients that resist to first-line osimertinib resistant is due to this MET amplification. So our goal was actually to use this forecasting to answer if we can actually anticipate this MET amplification before it occurs for selecting the patients who will benefit from upfront dual inhibition. And this is really important right now because there is this recent drug called amivantamab that it's able to target both EGFR and MET at the same time. The problem with this drug is that it's quite toxic. So it's really interesting if we can actually select those patients that will acquire MET amplification and therefore use this dual inhibition only in this subset of patients. For that, we started with in vitro data. We first tested our forecast approach using non-small cell lung cancer cell lines. All these cell lines have EGFR mutation, and what we did was to treat them with osimertinib for a long term until the emergence of resistant clones. The next step was to apply our forecast framework in this parental cell line and classify them as having high or low risk of acquiring MET amplification. And then what we did was to explore actually what we found in these resistant clones. And interestingly, only MET amplification was observed in the cell line, in the resistant clone of the cell line that we actually classify as having high risk of acquiring MET amplification. So this was really promising. And then the next step, what we did was to actually test our hypothesis of applying this dual inhibition upfront. So we take this high-risk parental cell line and we treat it for 30 days only with osimertinib to EGFR inhibitor or a combination of EGFR inhibitor plus MET inhibitor. After these 30 days, what we did was to perform single-cell DNA sequencing of these resistant cells to test or to check if the MET amplification was actually avoided. And this is what we see. If you can see in this plot, only the cells that were in the culture that was treated with only osimertinib present high copies of MET in the resistant clone, in the resistant cells. And we see a similar number of copies of these of the MET compared to the untreated one, and also in the bulk sequencing that we did in the parental cell line. So this upfront dual inhibition is actually avoiding the acquisition of MET amplification as a resistant mechanism. But of course, all of this work is done in vitro. So what we wanted is to see if that could work actually in the clinical setting. And for that, we collected a cohort of EGFR mutant non-small cell lung cancers that were treated at first line with osimertinib. The same, we applied our forecasting approach in this cohort to classify the patients as having high or low risk of acquiring MET amplification in the future. And what we see is that those patients that were treated with, that were classified as having high risk of MET amplification show a shorter progression-free survival compared to those treated with, classified as low risk of MET amplification. The problem here is that we don't have a biopsy of the relapse. And we actually cannot check if the patients that were classified as high risk of MET amplification actually acquired this oncogene amplification. So for that, we collaborated with the MSK to use the cohort of MSK lung cancers that were also treated with osimertinib at first line. Because in this case, they have data from the relapse. So we can actually check if those patients classified as having high risk of MET amplification acquire the MET amplification in the second sample. And our AUC values actually confirm that these patients that were classified as high risk of MET amplification actually acquired the alteration in the second time point. And for that reason, they have like a shorter time to treatment discontinuation compared to those classified as low risk of MET amplification. So just to summarize this second part of my talk, I show you that we have this forecasting framework that can anticipate amplification of driver genes that are important in the clinical setting. And actually, this framework might improve clinical decision making. But of course, our approach has some limitations. So one of the main limitations is that we are not able to know what are the processes that are actually active in a tumor sample and are causing the accumulation of new copy number alterations. And this has been my research line in the last 5 years. So, and we have used single cell DNA sequencing in order to try to solve this question. So just as you remember, we have this approach that it's able to take the different patterns of copy number alteration in a copy number profile to extract signatures, to know what is the biology behind those signatures, and to try to inform drug selection given these defective pathways. The problem here is with using bulk data, we don't have the information to dissect which processes are actually historical and has been operating during maybe the last 50, 20 years, even before diagnosis. And those processes that are ongoing, that are active, and are causing the accumulation of new copy number alteration. And disentangle this information, it's really difficult using, if not impossible, using bulk sequencing data. So for that, we have developed a framework that uses single cell DNA sequencing to know the dynamics of the mutational processes operating in a tumor. So what we do actually is to perform single cell DNA sequencing from the tumor cell population. We derive single cell copy number profiles of each one of these cells. And then what we do, our main point is to detect those alterations that are unique to each cell. Because our hypothesis is that those alterations that are unique are actually caused by an ongoing or active mutational processes that it's causing new copy number alterations. Those alterations that are clonal actually are caused by processes that are historical. And the last part of our framework is to assign this unique alteration to one of the signatures of our compendium of 17 chromosomal signatures, because then we can know which alterations or which different mutational processes it's causing the different unique alteration and are actually active in our tumor sample. So all this work was done because we have a specific hypothesis. Our hypothesis was that using ongoing chromosomal instability can actually improve drug response prediction. So it's better to tackle those processes that are active in a sample and are driving this tumor evolution and heterogeneity. So for that, We started with our BRCA1, BRCA2 deficient engineering models. We perform bull sequencing as single-cell DNA sequencing from the parental cell line, and then we treat them with PARP inhibitors and platins, so platinum-based chemotherapy, because if they are BRCA deficient, we expect to have that these cells are actually more sensitive than the BRCA1, BRCA2 proficient parental line. And actually, that is what we see. We see that both BRCA1 deficient and BRCA2 deficient cell lines or models are more sensitive to the wild-type counterparts that are represented in red. And these holes in the two PARP inhibitors that we tested, and also to the two platinum-based chemotherapies that we use in this experiment. So to test our hypothesis, we started with the bull sequencing data. And actually, when we use all the events observed at bull sequencing, we only detect a signature that it's linked to chromosome missegregation errors. So we don't have any signal of impaired homologous recombination in this parental cell line. However, when we use single-cell DNA sequencing and we limit all the signatures to those unique alterations, to these alterations that are actually caused by an active mutational processes, we have a high frequency of unique alterations that are mapped to CX3 and CX2 that are two signatures associated with impaired homologous recombination. So these differences in drug response can only be explained using this single-cell whole genome sequencing and our cell-unique detection workflow. To confirm this observation, we then move to a collection of pancreatic cancer organoids. We apply the same idea. So we sequence the parental organoids using bull sequencing data and single-cell sequencing data. And then we we treat those organoids with oxaliplatin, that is a platinum-based chemotherapy. And we see in our collection of organoids a range of responses. So we have organoids that are super sensitive to oxaliplatin and others that are very resistant. So then the next step was to apply our bulk data. So exactly the classifier that we use for predicting platinum-based chemotherapy resistant in our work that we published last year. And this is based on the ratio of two different signatures. So if the CX3 activity, that is the dark brown, it's higher than the CX2, the tumor is predicted as sensitive, and otherwise is predicted as resistant. So as you can see in the classification, our bulk approach can predict resistant this organoid that is actually the most sensitive organoid, and then predict sensitive this organoid that have high values of ED50 and therefore is quite resistant. So we moved to the single cell sequencing using only the unique events. And as you can see, in this case, we can actually classify correctly the organoids that are resistant to oxaliplatin and the one that is super sensitive as sensitive. So this actually indicates that detecting these signatures that are active or are ongoing in a tumor and are causing these new alterations can improve treatment predictions. So to summarize this last part of my talk, yes, I show you that we have developed this single cell sequencing framework where we detect unique alterations, and these unique alterations are linked to processes that are active in a sample. And therefore, the single-cell sequencing workflow can reveal clinical actionable ongoing processes of chromosomal instability that are not seen at pool level. So to just wrap up my talk here, I just want to summarize what I presented you today. So I started by showing you that we have this approach that is able to dissect the different copy number patterns or footprints seen in a tumor with chromosomal instability into signatures, and then these signatures can be used to predict treatment response. I also show you that we have this new forecasting approach, a framework that is able to predict or anticipate future oncogene amplifications and therefore can be used to predict tumor evolution. And finally, I show you that we have this single cell approach where we can detect those processes that are active in a sample. And this information is very valuable at the clinical level because we can tackle those processes that are causing new alterations in a tumor sample. And before to finish my talk, I would like to show you the work that we are focusing right now in collaboration with Sarah Aitken at Yale School of Medicine. I was there for 4 months and I'm still collaborating with them. And the idea is to predict tumor genomics using pathology slides, because the pathology slides are accessible in the clinical setting, and it's the first data that is coming from a tumor in a clinical workflow. And the idea is to use these pathology slides, apply machine learning and approaches in order to predict tumor genomics in these tumors. And in this case, we focus on liver cancer. So to end my talk, I think that this is the most important slides of all my presentation. This is a teamwork for sure. We know all of us that we are doing research, and I would like to thank my current lab at CENIO, also Icon Lab, because I'm like a researcher associated there, and it's really a pleasure to work in this group, all the collaborators, funders, and thank you for your attention, and I'm more than happy to answer any question.
Thank you very much, Bárbara. That was fantastic. A terrific overview of the work you're doing in your lab and in collaboration with Sarah. So we've got a number of questions which have come in, and we'll try and go through them. There are actually 2 from Ian Tomlinson, so we might take those one after the other. He did say, I'm not sure that you actually defined CIN at any point. And then I guess as a follow-on, can you actually treat CIN, and if so, does it work to make the cancer CIN negative?
Okay, so well, sorry if it was not clear, the definition of chromosomal instability. So what I was referring with chromosomal instability is these chaotic genomes with a lot of copy number alterations. So if you remember from one of my first slides, I showed this plot where the copy number profile looks like with a lot of points going on at that. I can try to share my screen. If not, again. Oops, sorry.
That's all right. We do have time.
Yeah. No, because this is a really important question. If it was not,
I mean, if it's not clear, then
yeah. So okay, I can use this one. Sorry. So the difference of a stable genome that has no copy number alterations at all, or very few. It's represented here in this plot where we have these chromosomes in the x-axis, so the different regions of the whole genome, and then we have the number of copies. So as you can see here, there is like no alterations going up and down. However, in samples that have chromosomal instability, you can see these huge complexity of copy number alteration. So there are regions that have 3, 4, 0, 1, and this is typically seen in tumors that have this chromosomal instability. So there is like a huge amount of regions of the genome that doesn't have the 2 normal copies that you are expected to see. And then the idea with treating these chromosomal instability tumors is more, rather than to go to a C-negative sample, is to kill or to kill those cells that have this huge amount of alterations. And normally, all the tumors with chromosomal instability are treated with this concept of synthetic lethality. So, for example, if a sample has deficiencies in repairing breaks, for example, in imperomotor recombination, what we do is to try to add a drug that increases the number of alterations or the breaks, so therefore the sample or the cell cannot correct those additional alterations, and it just cannot survive. So that's the main idea when we are treating tumors with chromosomal instability, to try to exploit those deficiencies in the pathways that they actually have.
That's great. Well, thank you very much for just clarifying that a bit further. So then a question, an anonymous attendee. So what proportion of chromosomal instability in human cancers can you explain by a specific underlying defect? It's a bit of a hard one.
Yeah. I mean, it's really difficult to say like a specific proportion or like a quantitative proportion. But it's true that normally it's an accumulation of different deficiencies along the way of this tumor life history. So, for example, you can start with imperomotor recombination because the patient is BRCA1, BRCA2 germline deficient, but then acquire like additional alterations that hits different drivers that start to to have deficiencies of miscegenation errors or in other pathways that are also causing this chromosomal instability. What is true is that we are, with this computational approach, that we are able to dissect the different signatures that are linked to different mechanisms. We can not only identify those, also to quantify what is the percentage of this tumor that is caused in theory by this signature. Of course, it's not a perfect quantification, but it's mostly giving you the idea of how much mutational processes is causing the huge amount of alterations that you are seeing in a profile.
Great, thank you. So Bharat Jasani, he's asked whether you could use your same technology in circulating tumor cells to predict therapy responses.
Yeah, I mean, I think that this is the same that we are working with pathology slides. I think that this is the other main point to go, because circulating DNA can give you like readouts of what is going on more real time. You can monitor better those patients. It's true that we have applied this original approach of identifying signatures using ctDNA in some samples of ovarian cancer. I guess that the most limiting point here is the amount of ctDNA that we can actually detect. And there is like a huge amount of work that it's being done by other groups to try to increase the amount of circulating DNA, or to actually apply all the different genomics technologies that we have been developing, not only in my group, like others, to try to monitor patients using these circulating DNA. And I think that this is the point there. It's true that we applied to ctDNA, but maybe we started with, I don't know, 100 samples. I don't remember exactly, but 100 samples, and then we get information from 30 of them, just because it's the ones that we have enough ctDNA released from the tumor and so on.
Okay, and just to follow up from Bárbara was to say thank you very much for your excellent, lucid and logical presentation. And you also get praise from Karen Orton, who says, great talk. In terms of translating this into the clinic, what would be the turnaround in gaining, or the turnaround time in obtaining such a CIN patient profile to stratify, in real time for treatment of the patient?
Yeah, I think that's a key question. So right now, with these biomarkers for selecting, for predicting chemotherapy resistant, we are starting a clinical trial here in Spain, where we are collecting retrospective data. So we did everything in a prospective way. So now what we are doing is doing everything at retrospective way. And actually, we are focusing on cancers that have high levels of chromosomal instability, but also that they are normally treated with chemotherapy as a first line because they don't have other options, like, for example, ovarian cancer, bladder. I think that there are some subtypes of lung cancer in the clinical trial. And the idea is to, with this trial, both two objectives we have. One is to validate our approach in a clinical setting, in a real clinical scenario. And second, to test that actually we show better responses in those that we have help on selecting the specific chemotherapy to give to them.
Right.
Yeah, and it's ongoing, at least.
So I have perhaps a follow-up question to that. Do you think that it would be possible in non-carcinoma cancers, so in, for example, melanomas or sarcomas, do you think this technology could also be applied there?
Yeah, so one of the tumor types that we tested in this publication was in sarcomas. Because sarcomas normally are treated with anthracyclines in the first line. The problem that we have with the sarcomas is that normally we don't have a standard of, like, an alternative standard of care. So all of them were treated with anthracycline. So what we did was to, rather than to emulate a phase 3 randomized clinical trial, we emulate a phase 2 clinical trial. And actually we see positive results. So and sarcomas have a lot of chromosomal instability. So I think that could be applied. The setting was not perfect to actually validate 100%, but yeah, it can be actually applied.
Okay. So Mark Aarons, he said, very impressive lecture. Thank you very much. How comprehensive is your classification of chromosomal instability mechanisms and processes? And are there CIN processes that are not fully identified as yet and understood?
Yeah, for sure. We have like some signatures that we don't know what actually is causing those signatures. This is quite typical in the mutational signatures field. Maybe you are more familiar with SBS, so point mutations in those signatures that are in this cosmic database. And there are a number of them that we don't really know what is the theology behind those signatures, but we see those patterns. In our case, most of those signatures that we don't know the etiology is because they're like really rare. So they're like really low amount of tumor samples that show these signatures of those patients. So therefore, we are not statistically powered to have a link with additional orthogonal data. And for that reason, we prefer to say, like, we don't know the theology, we need to explore more. Maybe if we expand the number of tumors now that we have like a lot of public data sets with whole genome sequencing around, we can actually expand and understand more those patterns that we don't really know what is the theology behind.
Okay. And last question, if you're still willing to answer them, is from George Seed, who says, thanks for the great talk. Is your synthetic clinical trial approach with the chemotherapy response prediction, did you explore other prognostic biomarkers? So how did the signatures compare in a multivariate analysis?
Yeah, so in this case, we like contextualize everything around this predictant resistance. So our endpoints for the clinical trials are for treatment response, not for prognostic per se biomarkers. And of course, all our analysis, for example, in the example that I show for taxane-resistant prediction in relapse ovarian cancer, we correct everything by other covariates that can actually explain this better survival of the ones that are predicted sensitive. So this is everything taken into account because there's like, and actually because it's real world data, so there is like a lot of confounding there. It's true that when you design a clinical trial from scratch, it's easier to take into account all these other covariates that can impact on your results. But yeah, for sure, everything should be corrected by stage, age of the patient, previous lines of treatment, for example, if it's not the first line, and so on.
Okay. And another question has just snuck in from Hugh Selway Clark saying, fantastic talk, thank you. Could some of the SIN signatures reflect selection patterns as well as incidence of SIN?
Yeah, that is one of the things that we have been also exploring. Because, for example, in this last part of my talk, where I talk about the cell unique alteration, it's true that these cell unique alterations could be because of the mutational process, or could be because they are actually more selected, because they hit specific drivers, and so on. So this is a thing that we have been exploring using computational simulations of birth to death simulations of clones that expands and so on, more in like in a mathematical way. But we need to actually then validate like in a in vitro control experimental setting, and we have not started yet, but it's a line that we want to explore in the future, because we think that this is like really key for these cases.
Okay, well, that's great. Well, thank you very much. Really appreciate, and I'm sure everybody appreciated your fantastic talk. In fact, you know, as somebody said, it was very clear, and you know, lucid and logical in
Thank you.
your scientific approach. And with that, I'd like to close this session and just let everybody know that we won't be having a grand round lecture next month in June because we have the Manchester Pathology or summer meeting, and therefore we will be taking a break in June, and similarly we take a break until September. And our next speaker will be Professor Trevor Graham, who was one of our keynote lecturers in the winter meeting in January this year. So with that, I'd like to thank you all very much for your attendance and wish you all a very pleasant and productive day. And thank you again, Bárbara. Goodbye then.